Welcome to all in your YouTube channel NCERT e-teaching decoding NCERT. In this video I am Dr. Kailash Mina going to explain chromosomal disorders in the category of genetic disorders from class 12th biology chapter 5 principles of inheritance and variation. This is our final video of chapter 5. So let's start the video. So in this video, after watching this video, class 12th biology students will learn about Down syndrome, Klinefelter syndrome and Turner syndrome. These all syndromes or diseases are chromosomal disorders. So let's learn how these type of disorders happens in human beings. In my previous video, I have talked about Mendelian disorders which were related to change in sequence of DNA or change in genes. So chromosomal disorders are the extension of that topic. So here the sequence of DNA will not change, the amount of DNA will change or the number of chromosomes will change. And I would like to mention one more thing, that in my previous video which was related to sex determination is very relevant to this video also, because you are having many clues about the mechanism of sex determination also. So if you have not watched those videos on Mendelian disorders and sex determination, you can check those videos out from the links given in the description below. Now here are few statements we need to clarify before we start those chromosomal disorders. Number 1, the chromosomal disorders, on the other hand, why we are using on the other hand, because previously we have learnt about Mendelian disorders. So on the other hand, are caused due to absence or excess or abnormal arrangement of one or more chromosomes. So there in Mendelian disorders we talked about the addition, deletion or changing in DNA sequences or sequence of nucleotides. Here we are talking about the whole chromosomes. They are deleting, they are adding, or there is some misarrangement. Failure of segregation of chromatids during cell division cycle results in the gain or loss of a chromosome or chromosomes called aneuploidy. See, here we learned one new term which is aneuploidy. What is aneuploidy? See, if you remembered the ploidy word from our class 10th science, class 11th biology and in the previous unit of class 12th biology, ploidy level indicates the sets of chromosomes in any cell. If any cell is haploid, then it will have one set of chromosomes. For example, human sperm and human egg in case of human beings, and all bacteria and fungi in other animals. If any cell have two sets of chromosomes, then this cell will called as diploid cell. For example, all cells of human being except gametes and all cells of other plants and animals. If any cell have three sets of chromosomes, then it will be called as triploid. We have encountered a triploid structure in case of angiosperms, and that was endosperm. We have learnt it in the chapter 2, sexual reproduction in flowering plants. And there are tetraploids, pentaploids and hexaploids also. You will learn in later chapters that wheat or Tetragum aestivum has 6 set of chromosomes. So there are many possibilities, but yes, you will encounter tetraploids, hexaploids, pentaploids, triploids in case of plants, not in case of humans and other animals, because the amount of chromosome or the number of chromosome is very crucial in case of humans and other animals. We cannot tolerate a single addition or deletion of any chromosome. You will learn later how it have serious effects on human beings. Now, so this condition of changing the number of chromosomes or in any human being is called aneuploidy. And why aneuploidy happens, and why or why these chromosomal disorders happens, why somebody have one more chromosome or one less chromosomes or two more chromosomes or two less chromosomes, because of the process of meiosis. As we have learnt in class 11th biology in the chapter 10 cell division cycle, and as we already have recalled this topic in my previous videos of principles of inheritance, that meiosis is very important in terms of genetics. So if you want to learn about gamete formation, if you want to learn about genetics, principles of inheritance or laws of Mendel, you need to recall the process of meiosis thoroughly. So I recommend you, if you have not recalled the process of meiosis, you must recall the process of meiosis before watching any of my videos of principles of inheritance. Now, so that set of chromosomes in any cell or in any tissue is called its ploidy level. So any cell is haploid, diploid or triploid or tetraploid, it is called its ploidy level. So this is what ploidy really is. Now we are going to learn about three syndromes as we have learned in the objectives, that are Down syndrome, Turner syndrome and Klinefelter syndrome. So here are a few summaries of those introductory summary. For example, Down syndrome results in the gain of extra copy of chromosome number 21. We humans have 23 types of chromosomes in case of females and 24 types of chromosomes in case of males. Of course, we have 46 number of chromosomes because we have double copies of all chromosome except in case of sex chromosome in human males. So, in general, we have 22 types of autosomes or 44 number of autosomes. So, the chromosome number 21 is among the shortest chromosomes and it will have very few genes. So, if we have one extra copy of chromosome number 27, it will result in some chromosomal abnormality which is called as Down syndrome. We will learn more about Down syndrome in my upcoming slides in this presentation. Now, similarly, Turner syndrome results due to loss of an X chromosome in human females. So, we have talked about gaining of one chromosome, that was chromosome number 21 in case of Down syndrome. Now, if any human female lacks one X chromosome, that she will have 45 chromosomes instead of 46 chromosomes. So, this condition is called as Turner syndrome, and these females are slightly abnormal. We will talk about this later in this presentation. Now, failure of cytokinesis after telophase stage of cell division results in an increase in a whole set of chromosome in an organism, and this phenomenon is called as polyploidy. So what is polyploidy? As I mentioned earlier, there is haploid, diploid, triploid, tetraploid, hexaploid. These conditions other than haploid and diploid are called polyploid. See, poly means many. So polyploidy is having many sets of chromosomes, and this condition or this phenomenon of polyploidy is observed more frequently in plants. Now, this condition is often seen in plants. We will not observe any of these ploidy conditions in humans or animals. The total number of chromosome of a normal human being is 46, 23 pairs, as we have discussed in this concept so many times. Now I hope you all have remembered this. Out of these, 22 pairs are autosomes and one pair of chromosomes are sex chromosomes. We have learned about this in sex determination also. Sometimes, though rarely, either an additional copy of a chromosome may be included in an individual, or an individual may lack one of any one pair of chromosomes. See, I want to learn one fact for I want to give you few concepts first, then I will read the letter statement. See, we have 23 pairs of chromosomes. It means every chromosome, either we are talking about chromosome number 1 or we are talking about chromosome number 5 or chromosome number 21 or sex chromosome, we have 2 members in each pair. It means we have 2 copies of each chromosome. So we are diploid for each chromosome. If we have one extra copy of any chromosome, let's say in this example of Down syndrome, we have one extra copy of chromosome number 21. So chromosome number 21 were 2 in number, which were 1 plus 1 in normal condition. That is why we are haploid. It means we have 2 chromosomes which are chromosome number 21, and if we have one additional copy, then chromosome number 21 will become 3 in number. So if we have one extra chromosome, it will be called trisomy. See, tri means 3. Why trisomy? We are only having one extra copy because we have already have 2 copies of that chromosome. Let's consider another example. See, if you have any chromosome, let's say again take example of chromosome number 21. If you have 2 chromosomes, 1 plus 1 in normal condition, and if one chromosome is missing, say it's one, if one chromosome is missing, then you will have only one chromosome. Then if you have one chromosome missing, then because you already have 2 copies of that chromosome, you will be left with one chromosome. So it is called monosomy. So mono means 1, tri means 3. In the same manner, if you lost the complete pair of chromosome, it means if you have lost all the 2 chromosome of any pair, then it will be called nullisomy. And if you gain 2 chromosome of any pair, it will be called tetrasomy. So why I am clarifying this? To understand the concepts. Why? Because if you have one extra chromosome, it will be called trisomy. Then there will be confusion in beginner's mind. Then why am I having one? Why this one is called tri? Because you have 2 extra already. And if you are losing one, then it will be called monosomy, because one will be still left in you, and one is called mono. And again, if you gain one pair, then it will be called tetrasomy. If you lose one pair, it will be called nullisomy. So these are very few simple general examples of having chromosomes extra or losing or your existing chromosomes. And let's consider another example, that if you have 2 extra chromosomes but not of the same pair. For example, if you have one extra chromosome of number 21 and another extra chromosome of chromosome number 20, chromosome number 20, let's say. So you will have 3 chromosome number 20 and 3 chromosome number 21. It will not be called as tetrasomy, it will be called double trisomy. So I am not saying that if you are having 2 more extra chromosomes, then you will have tetrasomy. If you have 2 more chromosome of same type, then you will have tetrasomy, otherwise you will have double trisomy. So this is the general explanation of this. You need not to remember all this, because only trisomy of 21 chromosome is included in your syllabus. So now you understand the terms trisomy and monosomy. This is what I want to take in your attention. So these situations are known as trisomy or monosomy. Now you know what is trisomy, what is monosomy. Trisomy, it means one extra chromosome. In case of Down syndrome, the chromosome was chromosome number 21, and in case of monosomy, as an example of Turner syndrome, where we have one X chromosome in case of females. These females have only 45 chromosomes. Finally, we are going to learn one more genetic disorder, which is called Klinefelter syndrome. And in this syndrome, again we have one more extra copy of one chromosome, and I am going to explain later what that chromosome is and how it is related to sex determination in humans. Now let's consider again this statement, because this is really very important. The statement number 5: failure of cytokinase after telophase stage of cell division results in increase of whole set of chromosome. See, if you want to understand this statement, you need to recall meiosis seriously. In meiosis, there is separation of chromosomal pairs during meiosis 1. Why? Because in meiosis 1, the subphase metaphase 1 includes chromosomal pairs arranged on the equatorial plate, not chromosomes as in mitosis. You can relate my statements when you are recalling the meiosis and mitosis. So I recommend you, while watching my video again, you must open your book or your textbook, textbook or notebook where meiosis is written, so you can relate these statements. So because of that, of chromosomal pairs at metaphase plate in meiosis first, the gametes or daughter cells are able to acquire single set of chromosomes, or we are able to have half number of chromosomes as compared to parental cell. So this is how normal meiosis happens. And what happens in abnormalities, that one of those pairs or more of those pairs, we are not taking the example of more because we are focusing on only one beginning idea. So one of that pair fails to segregate. It means if this pairs goes to one daughter cell, that daughter cell will have one more chromosome, and the other daughter cell in case of meiosis will have one less chromosome. And if this type of gametes made and survived, then if these gametes are going to fertilize with another gamete, then this will result in the abnormal chromosome number in case of human beings and in case of other animals and plants. So now you can have an idea why chromosomal disorders happens. Here we are discussing only what are chromosomal disorders. Now I gave you few hints, so it is the effort from your side, or you can say homework or assignment for you, so you can relate meiosis with these terms and you can understand how this chromosomal abnormalities happens now, and what is non-disjunction in case of meiosis. You can also learn, you can also research. I will take this topic separately after this chapter. Now our first chromosomal disorder, which is called Down's syndrome. So as I already have mentioned, the cause of this genetic disorder is the presence of an additional copy of the chromosome number 21, which is called trisomy. And now you know why it is called trisomy, because we are having 3 chromosomes in this case. And this disorder was first described by Langdon Down in 1866. The affected individual is short statured with small round head, furrowed tongue and partially open mouth. Palm is broad with characteristic palm crease. Physical, psychomotor and mental development is retarded. See, in this chapter, in this final video of principles of inheritance, and it probably the 13th video of the series, these chromosomal disorders are very easy to understand. And in these 3 disorders you need to remember the facts. So here the concepts are very less. As such, the characters of the disorders, you need to see the pictures given in your NCERT and need to connect and then remember those facts. Although I will try to connect other concepts from the chapter, so I am trying to make this concept more easier and more interesting for you by connecting different parts of the chapter. Now I am taking both of the Klinefelter syndrome and Turner syndrome in the same slide. Why? Because they have one connection. See, so one hint I was given you, that you will find what extra chromosome you will have in the Klinefelter syndrome. So here is the answer. You will have one additional X chromosome in males, and then males will become X and Y. So now you can ask one question also, and I am sure some of you are wondering why we are calling that this is a male with extra X chromosome, why we are not saying this is a female with extra Y chromosome. See, we have learned about the sex determination in human beings and in other animals in our previous video of principles of inheritance, sex determination. There we came to know that there is a mechanism which is called XX XY type of sex determination. XX XY type of sex determination, and the examples were human beings and the Drosophila melanogaster fly. And in this type of mechanism of sex determination, we came to know that females are homogametic and males are heterogametic. It means females are producing only same type of gametes, that are X containing, and males are producing 2 types of sperms, that are X and Y containing sperms. Now, and because of that, males were determining the sex of the child. Of course, there I gave you one hint that it on the superficial level, the sex determination in Drosophila and in human beings looks similar, but it is somewhat different in both of the organisms. How? I am going to answer that in this video. See, it is a normal condition where XX females in Drosophila and in humans and XY males in Drosophila and humans. But if any human have one X chromosome, then it will be a female, as in the case of Turner syndrome. I already mentioned that in Turner syndrome, female humans only have one X chromosome, that is why they are having 45 chromosomes. So this condition will be designated as XO. O means nothing, as we discussed XO mechanism in grasshoppers, as we have discussed ABO blood group mechanism. O means nothing. So if any person is having one X chromosome, it may be male, may be female, because it is if it is having only one X chromosome, it will be female, and if it is having one X chromosome and one Y chromosome, it will be male. Why? Because what we are studying, the Klinefelter syndrome and Turner syndrome here, are the actual reasons of the discovery of the mechanism of sex determination in human beings. See, these abnormalities are showing what is the responsible feature of maleness and femaleness. Here you can notice the presence of Y. By looking at normal condition, XX female and XY male, and by looking at abnormal condition, XXY male and XO female, you can say that the presence of Y makes anybody in human beings male, and the absence of Y makes anybody in human beings female. Although this is a very super simplified statement, there are many complications involved in that. There are many mechanisms, many factors, but yes, at this point of time you need to consider this: presence of Y maleness, absence of Y femaleness. But in Drosophila opposite happens. There XXY will be female and XO will be male. Why? I am going to answer that also, although this is not related to this topic, but it will promote in you the critical thinking to look at sex determination and the chromosomal disorders, and it will be very useful for you also to remember these facts, and it is definitely going to help you in the NEET examination also. So this is the difference in Drosophila and in human beings, that normal XX will be female in both Drosophila and human, normal XY will be male in both Drosophila and human. But in case of abnormalities, XXY will be male in humans, XXY will be female in Drosophila, XO will be female in humans, XO will be male in Drosophila. Here you can see the connection, that number of X chromosome, whether they are 2 or whether they are 1, is determining the sex in Drosophila. So in Drosophila, if you see the normal condition, 2 X chromosome means female, 1 X chromosome means male. But in human beings, absence and presence of Y chromosome is making someone male or female, and that is why XX doesn't mean male, XX doesn't mean female all the time, because you can see that XX is female here in normal situation and XXY is male here in Klinefelter syndrome. Why? It is male not because of number of X chromosome, because of presence of Y chromosome. So why XO is female in case of humans? Because of absence of Y chromosome. And why XO is male in case of Drosophila? Because of one number of X chromosome in normal condition. So superficially, when we see normal things, then Drosophila and humans appears the same, and they are having the same mechanism of sex determination also. But the complexity is somewhat different. So we learn about, or scientists learn about, the mechanism of sex determination, which we have studied in my previous video and in NCERT textbook. Because of these disorders, because of these abnormalities, they came to know, okay, that this thing may be playing some factor in the mechanism of the sex determination. Then they will discover by performing several experiments, trial and error methods. Now I am concluding this statement by giving you final hint that in Drosophila, the ratio of X chromosome to their autosomes determines the sex of the individual. Although this mechanism is not mentioned in our NCERT textbook in that much of detail, although it is mentioned in our NCERT textbook, but not in that much of detail. but you need to keep in mind, because we are going to face another examination also, which is NEET examination, and the curriculum of CBSE is also revising. So it may be included. You can see the appendix in your new NCERT textbook, or you can always see the latest curriculum of CBSE. Now, in the Drosophila, the ratio of X chromosome to number to the sets of autosomes. For example, if Drosophila is diploid, so that is 2. Number of X chromosome in male will be 1. So 1 divided by 2 is equal to 0.5, and in case of female, it will be 2X chromosome divided by 2. What is here? 2 number of X chromosome. What is 2 here? Sets of chromosome. Again it will be 1. So if this ratio will be 1 in case of Drosophila, it will be female. If ratio will be 0.5, it will be male. If ratio will be 0.67, it will be intersex. If ratio will be more than 1, it will be super female. Less than 0.5, it will be super male. So here I am giving one idea. If you are interested, then you can search by your own, by various means, the mechanism of sex determination in Drosophila. Then you will come to know that it is not that simple as we have thought previously, the XXXY mechanism. Now coming back to the topic, Klinefelter syndrome and Turner syndrome. So here you need to remember the characters, the abnormalities as such, by associating with diagrams which I have taken as it is from our NCERT textbook. So here you can see that first, taking the example of Klinefelter syndrome, this genetic disorder is also caused due to the presence of an additional copy of X chromosome, resulting into karyotype of 47. So we have encountered 47 number of chromosomes in case of Down syndrome, also in case of Klinefelter syndrome. But yes, in Down syndrome the extra copy we are having was chromosome number 21, and in this we are having one extra X chromosome in any male. So such individuals has overall muscular development. However, the feminine development of breast, gynecomastia, is also expressed. So these are abnormal males. That is why diagram is also given, so that you can visually remember those characters. Such individuals are sometimes taller than average, always sterile, and always male. Why they are always male? As I have cleared, that because of the presence of Y chromosome. Now, taking the example of Turner syndrome. What is Turner syndrome? Such a disorder is caused due to the absence of one X chromosome, that is 45 number of total chromosome, and this condition is termed as XO. Such females are sterile as ovaries are rudimentary, besides other features including lack of other secondary sexual characters. So these are abnormal females, as you can see in diagram also. Now, it is the only known case in which a person can survive with only one member of chromosome, here XY pair. Or you can also remember that XO were males in case of grasshopper also. Now, here I have pointed out this statement also, which I have discussed in case of sex determination. These observations suggested that the gene that determines maleness is located on the Y chromosome, and here is the name of that gene, which is called as RY, the sex determining region of Y chromosome. So this region or this loci is located on Y chromosome, which is a DNA which will transcribe into RNA, which will make proteins, as I have discussed in the topic mutation and pedigree analysis in my previous video, and they will result the maleness. It is very complex process. Now, so it is the only known case. See, this statement is also very important. Only known case with only one member of chromosome. Why? Because the fact is, we humans or many animals cannot tolerate extra amount or lack of any chromosome or DNA, and that is why any change in the amount of DNA results in these disorders. You can see now that only a small chromosome, which is Y chromosome, or small chromosome, which is 21 number chromosome, small chromosome, which is X chromosome, these chromosomes are having very few number of genes, and the absence or presence of these chromosomes results in that much of abnormalities. For example, 21 number chromosome having very few genes, and that is why we are able to see these type of individuals. You cannot see trisomy of chromosome number 1 or trisomy of chromosome number 2 because these are very big chromosomes having large number of genes. So here is one another idea which is called doses compensation. You can search by your own by to feed your curiosity that we humans cannot tolerate the abnormal doses of chromosome, and that is why although females are having 2 X chromosomes and males are having 1 X chromosome, and still females are having only 1 functional X chromosome, and the another X chromosome will be inactivated in the form of heterochromatin, and that inactivated X chromosome is also called bar body. So this is also a homework for you to search about bar body. So bar bodies, I am telling you, are the inactivated X chromosomes in the cells of females, and because of that you can also determine that which cell belongs to male or which cell belongs to female, because female cells will have a bar body and of course male cells will not have a bar body. What is bar body? It is an inactivated chromosome. So here are many concepts which you can search in your textbooks, in your with internet, and you can increase your knowledge with your curiosity. So thank you so much for your patient listening. Here I am concluding this chapter number 5, and in my next video we are going to begin the chapter number 6 of class 12th biology, which is molecular basis of inheritance, and we are going to discuss the topic DNA. Thank you so much.